Welcome to my talk about connecting Shiny apps to Chemotion-ELN. I'm a postdoc at the Karlsruhe Institute of Technology, working in the group of Professor Krämer, the so-called IBCS FMS. We work on questions regarding or addressing chemistry, but also biology. And first of all, I want to give you an overview of my talk. I will begin with an introduction to NFDI for Chem and the Chemotion-ELN. ELN is an abbreviation for electronic lab notebook, and a lab notebook is traditionally, it's a book where scientists write down their documentation of their experiments, but also describe what they found out in the lab. The second point of my talk will be how we extend Chemotion-ELN with Shiny apps. And as a third point, I will show you an actual example of a Shiny app, which is an extension of a Chemotion-ELN, so-called Biostats app, which is an app useful for biologists who want to analyze and interpret the data. So especially visualization is supported, but also a bunch of statistical tests. So let's get started with the introduction to NFDI for Chem. It's a consortium which is located in Germany, and if I would translate it roughly, I would say National Research Data Infrastructure for Chem. So for chemistry, there are also other groups which support biology and other subdomains of it, but we are responsible for chemistry, and our goal or aim is to build infrastructure for scientists, and so mainly software, so that they can efficiently analyze and document their work. And it is also important for us that the data which is produced in the lab or as a theoretical simulation, for example, is fair. Fair is an abbreviation, so findable, accessible, interoperable, and reusable. And we hope that in the future, almost all data will be fair. For NFDI, as I said, is a consortium located in Germany, and many universities and research groups are within the consortium and work on the infrastructure. For chemists, as I said, I work at the KIT in the southwest of Germany, but we have many other groups involved in it. But our group is, or we have a main core developer group for the Chemotion-ELN. To make the motivation clear, I just want to go a little bit into history. During the last century, computer become more and more available. So at the beginning, there weren't computer at all, but in 1985, the supercomputer Cray 2 had already 1.9 gigaflops, and this is pretty. This increased pretty much because in 2017, a common iPhone 8 had already 325 gigaflops. So we have an enormous amount of computational power nowadays. In contrast, when we look how scientists document and analyze their stuff, sometimes it didn't change a lot, especially with the documentation. You see on the left bottom side a lab notebook from 1927, and basically the scientists write down what he or she wanted to do in the lab and what they find out. And you see on the right side in a lab notebook from 2023, and it looks almost the same. So it's also handwritten and describing what they want to do in the lab and so on. So here, this is really a traditional workflow, and but we want to offer more modern workflows to get a better documentation and ensure that the data which is produced is fair. And therefore, I want to describe certain areas which are involved in for this modern workflow. First of all, the device integration. So, for example, a chemist measures a sample on a HPLC, and the HPLC produces a chromatogram. And previously, the scientist goes to the device, get the file with the stick or whatever, and analyze it on its personal computer. Nowadays, or in our lab, the data is sent directly from the device to the ELN. Within the ELN, it is linked to the sample, so the actual planning of the experiment, so that it is well documented and the scientist doesn't have to do anything with it. Beyond the device integration, we are working on different software developments to analyze data, especially in the area of chromatograms. The main software, which is the electronic lab notebook, it's mainly focused on chemistry. We have also generic functions. But as I said, it supports special fields like organic chemistry, physical chemistry, inorganic chemistry, and so on. I will show you later 3 features of the ELN, which the ELN can do. And as I showed you in the last slide, this approach to scientific documentation is pretty new. And therefore, it is necessary that this is also teaching this new kind of workflows, and the software has to be taught to scientists so that they accept it and they use it in their normal workflow. So that is also an important point, which is done by the NFDI. And the last point, of course, for documentation, but this is mainly related to software development. It is not only developed, but it is also really used for ELN. And as you can see here in green, that's a map of Germany. Many universities are already using it. And not only in Germany, but at least some of it are interested in other countries or are already using it. So the basic idea is that each working group has its own decentralized ELN. So they have some data which they don't want to share. It's on their server and nothing happens. But if if a paper is published with data, then this data should be fair, so it should be available to all, and therefore the data is collected in so-called central repositories, like GitHub, basically, for idea. Here we have data collections of published data, and everyone around the world can access the data. And now I want to go one step further and show you 3 features of Chemotion-ELN. First of all, so this is the website. If you open it, you have on the left side so-called collections. A collection is basically a folder, and but you cannot put a folder in a folder here. You have only one level of depth, and within one collection you can place different objects. For example, here the chemist has put a bunch of reactions. So the object here is called reaction. And we see here one reaction in detail on the right side. So, so this is the plan of a chemist you want to do in the lab. So one possibility is to plan really chemical reactions. This is especially interesting for the organic chemists. And if the experiment is done in the lab, then the chemist knows how the amount of the product he gains and the purity and so on. And all of this characteristica he can document now within the ELN at the same place where he planned his reaction. Besides planning this, it's also possible to draw structures, which is also a pretty important point for chemists. And here we have 4 different softwares. 2 of them are free on the left side, and on the right side, we have commercial ones. Catcher Rails and Catcher 2 on the left side are already integrated into the ELN, and no, only Catcher Rails. Catcher 2 is only partially integrated, and full support will come. But as you can see, compared to the commercial one, the resulting molecule looks already almost the same. And also for features which are available to draw a compound are comparable. So this is pretty cool. As a side note, our software is of course open source, and later you can also see the link. So if you are interested, you can go on GitHub and see the entire software. Besides drawing structures, another important point if the experiment is done is to analyze the data. Here I've brought 2 results. One is the result of cyclic voltammetry data, and one is size exclusion chromatography. as you can see, it's not only visualizing the data here, but what it can be also done more. So certain properties of a curve can be analyzed, and this is necessary to interpret the data. And yeah, you can see it also on the right side. It's not only for visualization, but you can also conduct so-called peak picking. You basically say where peak starts and where it ends, and you get the area under the curve, which is important because it's proportional to the amount of the compound you have in your sample. So this is the analysis of the data. The software for ELN is pretty large, and therefore it is difficult to integrate all of the possibilities to analyze the data. And therefore we thought it would be really cool if we could, if the ELN could communicate with other apps. And that's the next topic of my talk. So we have here, for communication, we have here the ELN on the left side. And what I understand with under communication is sending files between the ELN and other apps. And these other apps can now be Shiny apps, for example. I will show you now a little bit what goes behind the scenes. So let's say we have scientists, and finally all the measurements are done. A bunch of results are on the computer at the ELN. And now the scientists want to analyze and interpret the data. So I press send file, so it should be sent to the third-party app. But first of all, the ELN front end, which is written in React, has to communicate with the ELN backend, which is a Ruby on Rails project. The backend creates a token, which is used for verification and holds some information about the user and the file and so on. And this token is used now by the front end to assemble a new URL, which consists of a URL of the Shiny app or for other third-party apps. Doesn't have to be a Shiny app. And as a query parameter, the URL of the ELN itself is added and the token. Now the Shiny app is opened in a new tab in the browser. And first of all, the Shiny app conducts a GET request at the ELN backend. And this is necessary to download the file. So it has now the file, can load it into R. The scientist can now do its analysis and interpret the data, visualize the data, whatever he wants to do, or she. And afterwards, he says, okay, send it back to the ELN, and the Shiny app now saves all of the results of the analysis again in a file and conducts a post request so that the file containing the results is now on the ELN backend. The cool thing is, if you write the Shiny app, you don't have to care at all about all of this stuff. So you, as a mental model, it's okay if you think about the ELN and the Shiny app by sending files to each other. There are basically, I have written a tiny R package called ComELN, so Communication-ELN, and it has two functions, download and upload. If you want to get a file from the ELN, you use download, you pass the session object, which holds, among other things, the URL, and you say, okay, please download the file in the folder, which is the second argument, so a folder path has to be added. And if you have done your analysis, you store the entire stuff in a new file, and you use the upload function, pass the session object, the file, which is called f here, and you pass also a new name. The new name is basically the name of a file which will be used in the ELN. And that's pretty much all you have to know if you are writing Shiny apps, and thereby it is possible that scientists who can code can provide their analysis by writing a little Shiny app. And this is pretty important because not so many scientists can code, and thereby it gets publicly available. And now I want to show you the actual app I was talking about, the so-called Biostats app. First of all, I have here the ELN again. So we have here two collections, which are banana research and CO2 uptake. Now CO2 uptake is chosen. From CO2 uptake, there's a research plan. A research plan is basically like a you write text and describe your experiment, but it holds also attachments, and from the attachments you can here have a drop-down menu, choose the app, and send it to the. Now a new tab is opened, and the Biostats app is you see here. So let's go one step further, and see an overview of the entire app. So you have a data tab where you can calculate different operations. You have correlations like Pearson, Candle, and so on. Visualization, I will show you in the next slide. You have box plots, scatter plots, line plots, and many other features. Then you have an assumptions tab where you can calculate Shapiro test, Levine test, you can do this diagnostic plots and so on. So this is pretty cool, especially for biologists, because often biologists only do t-tests in Excel, which is a pretty bad thing. And it's often the wrong test, which they choose, and they didn't test at all the assumptions for it. But for yeah, so this is cool if they can do it, because the main hindrance is that often they cannot code. And here is the test tab, which has a t-test, ANOVA, and so on, and also has a lot of post hoc tests like 2K-HSD and so. And these tests are actually not available in Excel, so it's cool for them that they can use it now. And as you see, you have here the dependent and independent variable, which is basically the same if you write a formula in R. Yeah, exactly. So now you have seen how to do a 2K-HSD, and there as a last possibility there is so-called dose response analysis, which is a pretty common experiment in our lab. Now I want to show you the visualization. I use ggplot as a backend. So yeah, in the backend. And yeah, here you see again, you can calculate some operations. Can, for example, if you have the wrong unit of your data, so you can fix this. And visualization, as I already said, we have box plots, scatter plots, and line plots. You can add uptake and concentration here. When you say, okay, it should be numeric, you can add the labels. If you are familiar with ggplot, you see that it's basically a ggplot or part of ggplot in a UI. Yeah, you can also, if you, so now you have different color themes and fill themes. And now I go to the scatter plot, as you see, and here you can also fit with, for example, here a generalized linear model. And I added the formula and so on of a model in the plot, which is quite useful. And as you saw, you have saved it, and the file was uploaded successfully to the ELN. If I go now one step further, and we are now back in VLN, you have to reload your website. And when you have done this, when you go back to the attachment tabs, and here we have a new file. So the data is now available within VLN, and new file contains, in this case, the plot. So yeah, as an additional information for our package, COM-ELN and Biostats are available on ComPlat ShinyChem on GitHub. So if you yeah, you can see the code and test it, whatever you want to do. And what is also cool, Biostats is also available as a standalone serverless version using Shiny Live. And I have here the URL, which is a github.io page of the same repository as above. So you can use ShinyChem, so not ShinyChem Biostats. And yeah, I hope that scientists will find this useful. And for serverless version, of course I don't have attached it to the ELN, but it has an upload and a download button, and the results are in a zip file when downloaded. So this is a really standalone and don't need an ELN. And with this, I come to an end. I would thank, of course, the NFDI for Chem for funding. I would like to thank the entire development team and our entire research group. Special thanks go to my boss, Nicole Jung, who encouraged me doing this and helped me a lot. I would also like to thank Pierre, who helped me a lot with coding and setting up the ELN and so. And I would also like to thank my colleague Martin Sterman, which is not here on a picture, and he helped me a lot during coding and implementing this in VLN. So with this, I come to an end. I would like to thank you for listening and for your attention, and I would be happy if I could answer all your questions.